All right, class. Last time we did discuss about the anatomy of the eye, and we did manage to go through all these parts. So we did discuss the parts which are sensitive to light, and also discuss about the parts which help facilitate interpretation of light energy, or the conversion of light energy from light into chemical, then also to change it into what we call electrical. Then today we are going to be continuing with the same anatomy and some of the parts and how cardinal they are in terms of vision. So last time we also looked at the fluid field components. So the retina is flowed by jelly-like fluid known as the vitreous humor, okay, which we talked about. I believe last time when we learned, we talked about where we can find the vitreous humor. Okay, so the vitreous humor is this part. Okay, then there's also what we call the aqueous humor. Then it goes up to the ciliary area and is made up of elongated proteins and is not freely flowing, meaning this part does not flow freely. Anterior to the lens of the eye is the aqueous humor, which is freely flowing. Okay, so like on behind it flowing, and other pathological conditions that come about if it's not controlled in terms of production of the aqueous humor, etc. Then both fluids can maintain the structure of the eye and intraocular pressure, which is about 10 to 20 millimeters of mercury. So if you look at this range, 10 to 20 millimeters of mercury, this is the right amount of ocular pressure that is needed. To be more precise, intraocular, because if this pressure increases, it might lead to other pathological conditions. Then the aqueous, the humor also provides nutrition to the vascular area and gets rid of the waste metabolites. So when you say the humor also provides nutrition, meaning it's able to supply nutrients that are cardinal and important to the cells that are found inside the eye, okay? And also to the vascular area. What do you mean a vascular area? This is an area that doesn't have blood vessels. So this is able to supply these areas which do not have access to blood vessels. So it's able to provide nutrition, meaning that the humor is highly nutritious and also able to get rid of what we call waste metabolites. Okay, then when you go on slide number 6, we are going to look at the different, we're going to look at what type of fluid is produced in the front part, or we can say in this cavity where we have a fluid known as aqueous humor being produced. Okay, so if you can see, the title is saying production and circulation of aqueous humor. It's just an abbreviation, AQ, simply to just show us that we're talking about aqueous humor. Okay, so now when you look at this slide, you're going to realize that it is produced by what we call the ciliary body. Okay, so now if you finish the first introduction to the anatomy, we are still talking about the anatomy, but the first part of the session, we were talking about the compartments of different parts of the eye. So this is the lens, and if you know, these are ligaments, especially ligaments, and these are known as the ciliary bodies. They're found on the right and the left. Then there's a pupa, which is the space in between two iris. Okay, it's a space because the iris forms more like an oval or a round shape. So the space here is known as the pupil. Okay, then this is the cornea. Okay, then there's also the anterior chamber and the posterior chamber, what you can see there. And then there's the sclera. Okay, yeah, so the sclera and the cornea, remember in our first part we talked about the cornea and the sclera being the fibrous tunic, meaning they're the ones which are responsible for protection. Okay, so now allow me to go into the details. So the same ciliary bodies has what we call the stroma and the epithelium that play a major role in the production of the fluid. So if you want to understand how this is produced, okay, you have to know that it's the stroma and also the epithelium that have good cells that are able to, that have good cells that are able to produce what we call the aqueous, humor. So now the epithelium absorbs the sodium from the stroma within the ciliary body and secretes it into the ocular lumen. Okay, so lumen simply means it's a space, an opening that is found in a certain area. Like for instance, it might be a tissue, or it might be something. Okay, for instance, I'll give an example. If you talk about GIT, when you look at the olo, or the inside part or structure of the intestines, that's known as a lumen. Okay, even the blood vessels. Okay, the empty, I can't say empty because there's something found there. It's also known as it's also known as a lumen. So we have got what we call the ocular lumen. So the ocular lumen, that's where when we produce this fluid known as aqueous humor, it's going to go into this ocular lumen. Okay. There is a flow of chloride and bicarbonate that allows an osmotic pool. So if you understand cell physiology, you can appreciate what we mean by osmotic pool. Okay. So osmosis has to do with just diffusion, meaning where there is a higher concentration of solutes, meaning that's where the fluid will be going. Okay. So in this area, you'll be moving from where we have got a lot of water or fluids, okay, then going to where we have got less amount of water, where there's more concentration of solutes. So that's basically osmosis. So other secrets include, so other substances which are secreted includes the ascorbic acid, glucose, and amino acids. Okay, so if you look at all the chemical nature, we go back again to cell physiology. No wonder I always tell my students that in order for you to understand and appreciate physiology, you need to master cell physiology. Okay, so now when you look at all these electrolytes, okay, glucose, sodium, molecules, and electrolytes, bicarbonate, they create what we call an osmotic pool. Okay. So now, about 2.5 microliters is drained daily and circulates to the interior component of the eye. So this is the amount that goes to the ocular lumen, inside the space that's found in the aqueous, the space which is found between the cornea and the lens. So you find that there will be a fluid which is going to be there, which is known as aqueous humor. And the amount is 2.5 microliters. Is drained daily and circulates to the interior component of the eye. So this is how this space is filled with a fluid. But again, if there's too much of a production of aqueous humor, it leads to a certain condition known as glaucoma. Okay, it leads to a condition known as glaucoma, and we'll talk about this as we go on. I'm sure you guys will have come across this, and we did discuss what glaucoma is. And I was saying glaucoma is a condition whereby the front part of the eye or the cornea appears to be clouded. Okay, that is known as glaucoma, and it's more common in old people. Okay, so now we are still the same diagram. Please don't think that we are talking about something else. So now there's a meshwork of tissue. A meshwork of tissue is present around the cerebral body to drain the fluid and maintain pressure. Okay. So a meshwork of tissue is present around the cerebral body to drain the fluid and maintain pressure, because pressure is very important in the human eye, so that we prevent eye defects. No wonder if you look at the pressure range, we said 10 to 20 millimeters of mercury. Okay, so this is known as the Canon of Scalar, which with the tubercular meshwork. The meshwork has the fluid passing through it into the canal. The TI has collector channels that join to form aqueous veins that drain into the episcleral vein. Episcleral vein, meaning it has to do with the sclera. With age, the meshwork becomes sclerotic. Okay, sclerosis has to do with hardening. Okay, when you say someone has got sclerosis, it means there's hardening of a certain part of a certain tissue. So that's what they mean when they say with age, the meshwork becomes sclerotic and may result in drainage resistance, leading to glaucoma. This means that when the meshwork becomes sclerotic, meaning it becomes hard. Okay, it's the same thing when you are building something. Maybe you are creating a foundation. In order for it to prevent a fluid from passing through that building or something that you are raising, the concrete has to become hard. But if it's soft, there will be a continuous flow of the fluid. Okay, so now with time, even most conditions, when you talk about BP, high blood pressure, causing sclerosis in the renal veins, and in the renal blood vessels, ABCD, all those things that leads to either acute or chronic kidney failure, all those things has to do with the damaging or the hardening of those structures or areas. That's what we are talking about. So with age, the meshwork becomes sclerotic and may result in a drainage resistance leading to glaucoma. And glaucoma is very common in old people. No wonder if you find that with age, I'm sure maybe the meshwork becomes sclerotic when people grow older, or maybe they're pre-exposed to certain conditions that result in this same phenomenon. All right, now what you're seeing here is application, okay? What you're seeing here is application. Debris in the fluid may cause clogging, leading to the secondary glaucoma. The system is kept free of such eventualities by the presence of another drainage system through the ciliary muscle to choroid and then the suprachoroid space, okay, the suprachoroid space. The meshwork also have the microphage that helps clean the fluid. As if you don't know what a microphage is, they are simply white blood cells. They are phagocytes, okay? Meaning they're able to digest any foreign particles that might cause the blockage or clogging, okay? So angle problems lead to pressure buildup, resulting in degradation of the retina. Hence, it leads to impairedness in terms of, it leads to blindness, okay? So angle problems lead to pressure buildup and result in degradation of the retina. Remember, the retina is the area which is sensitive to light. Okay, so this diagram, they're just trying to talk about two different types of glaucoma. So if you can see, the first one is known as open angle glaucoma, and the other one is known as closed angle glaucoma. So the blocked drainage of aqueous being shown here, okay? Arterial chamber open, and then the blockage at the trabecular meshwork. Then there's under closed angle glaucoma, there's blockage drainage of aqueous, and there is also anterior chamber angle closure, okay? So these are the two different types that we can at least talk about. So now let's go to slide number 10. So here we are just trying to talk about the anatomy, the internal anatomy of the retina and what is found there, okay? But on the glaucoma, please go and read that up, find enough information in books that can help you facilitate to understand what we mean by open angle and closed angle glaucoma, okay? So that you are not left out. So now let's look at the retinal features, okay? So when you look at this diagram, okay? When you look at this diagram, allow me to increase the font, okay? So now, here we have got this part. So as you remember, the sclera was part of the fibrous tunic, if you remember very well, okay? Then we had this structure, which was known as part of the retina, okay? In this part of the retina, that's where we had this is the choroid. Forgive me about that. This is the choroid. So there's the sclera and there's a choroid. So the choroid and the sclera, they were just part of what you call the fibrous tunic, if you remember. But anyway, when you talk about the choroid, what do we mean? This choroid is also responsible for absorbing UV rays, okay? That's one of the functions. Because ultraviolet rays are very disastrous to our health. Because when you look at even melanin, yeah, that's what it does. There's what we call melanin quenching, which has to do with absorbing or trapping these UV light, okay? So now, then you can see that there is a layer of different types of cells, which you can be able to see there. So this is the choroid, then there's the sclera, the choroid. Then what you are seeing here, which is just coming adjacent after the choroid, these are known as the retina. And the retina contains specialized cells, okay? The retina contains specialized cells, okay? And these specialized cells are responsible for trapping or for absorbing light rays, okay? So now, when you look at this, we have got what we call the cone cells, and we also have got what we call the rod cells. The cone cells are responsive to bright light, or light that has got has got high intensity, while the rods are responsive to light which has got low intensity, okay? So now when you look at this diagram, you're going to see that we have got here, okay? Here we have got what we call the peripheral retina, okay? This is known as the peripheral retina, which you're able to see. Then we have got, these are rod cells, okay? And we have got cone cells. So if you can see the way they are positioned The cone cells are found on what we call the fovea centralis. No wonder we said during accommodation, if light falls on the fovea centralis, meaning an image which is clear is going to be depicted. Whereas if it falls on these other parts where we have the rod cells, the image will be very poor. Then here we have got what we call the optic nerves. Remember, like last time what we learned, the optic nerves, this is where we have a blind spot because this is where the optic nerves leave the eye, okay? And the optic nerves, just like we talk about nerves, nerves carry what we call electrical energy, okay? So in form of impulses, so they'll be living here. So here we are not going to have any image that is going to be captured, okay? So don't worry about these numbers. So what these numbers mean, so if we have got what we call here the epithelium, okay? Then if we start from here, light first goes and strikes or goes and strikes what we call number one. So these are the rod cells. So in dim light, like in the dark, okay? It's very sensitive to dim light, okay? So now we are going to find that even one photon of light can be able to stimulate the rod cells. So these are the rod cells, they'll get stimulated. Then after that, they're going to pass on to what we call the bipolar cells. From the bipolar cells, they're going to pass on information to what we call the ganglion cells. So remember, in between here, we have got what we call synapses. Remember, synapses are junctions that are able to communicate or create a communication or send messages between two adjacent cells, okay? By converting chemical into electrical energy and vice versa. That's the way it is, okay? So that's what is happening here, okay? So now, after the ganglionic cells, then they're going to transport via the optic nerves or optic fiber to the eye so that the image can now be interpreted in the right pattern. Then when you look at this diagram, there's one funny thing. If you look at it, they have given a color which is pink. If I'm not colorblind, you might never know because most men are colorblind. So if this is pink, you realize that this is known as a cone cell, but we have got also a cone cell. So just like the last video that we had, I was explaining that when you talk about the cones are not only found on the fovea centralis, they are also found spread around the retina, but a higher concentration is found on the fovea centralis. No wonder we get a better and clear image. So another thing that you're supposed to know is that when you talk about the sensitivity, this is more, it requires a lot of stimulus, a lot of light for them to be stimulated, the cone cells. We'll talk about this in detail later. So now we're going to find that there's 1, there's 2, and there's 3. So photoreceptor mainly known as the cones, then the bipolar cells, then there's 3, the ganglionic cells, then later it's transported by the nerves to the brain for interpretation. So basically this is what the diagram is talking about. But the funny thing, like I told you, we need to understand first the anatomy. So these diagrams will be becoming more and more sophisticated as we move on. So no wonder it's very important for you guys to appreciate the information as you move on. So if you didn't see the last video, please go back and see the first video on the anatomy of the eye. Then come to this video because in the next few slides, we'll go deeper and deeper so that you can be able to appreciate the information. All right. So now the retina, the light sensitive part of the retina moves along the second layer. It extends anteriorly almost to the cerebral body. Looking through the scope, 2 points are shown on the retina. Okay. The macula and the optic disc. The posterior has a yellowish area with a nasally placed optic disc. Okay, the yellow part is the macular disc, which with a slight depression at the center with a sharpened vision, known as fovea centralis, the one that we were talking about in the previous slide. Okay, that's the fovea centralis. If you are unable to see it, this is where the fovea centralis is. Okay, so now it can be differentiated by color, placement, and also vascular. So meaning the type of blood vessels which are there. So this is what just they're trying to talk about. So there's anteriors, venous, then there's also what we call optic disc. Okay. All right. So now the arteries enter the optic nerve. Actually, let me just go back to the other diagram. So now when you look at this, that's the fovea centralis, then there's the macula, retina, and yeah. So this is just showing an external part, trying to show the blood vessels and where the fovea centralis is, etc. So now let's go to this other slide. So now the arteries enter the optic nerve to the optic disc, divided into two branches. This makes the optic disc highly vascularized, meaning has a lot of blood vessels supplying the inner retina. There are no rods or cones in the optic disc due to the nerves going through it. The center is known as the blind spot, correct, which I've talked about, and the light is therefore focused on the macular retina, which has a center of highest number of cones. Okay, that's what they're showing on the diagram. So like I said, these structures become more and more complicated. So go through what we learned, and next time when we meet, we are going to look at the layers of the retina in detail so that we can be able to appreciate the information. All right, so this is the end of my lecture. Until next time.